So I want you to picture just a massive neon-colored coral reef. You're hovering right over it.
Oh, yeah, just teeming with life.
Right, exactly. It's this bustling underwater metropolis.
Yeah.
Millions of fish, constant movement, just incredibly vibrant. But biologically speaking, it really shouldn't exist.
No, it shouldn't. It defies logic.
Yeah, because you're looking at one of the most productive, energy-dense ecosystems on the planet, but it's thriving in ocean waters that are basically, well, a nutrient desert.
Right, a complete void of nutrients.
It's like finding this sprawling high-tech city that operates entirely on solar power, but it's built in a total vacuum. It just doesn't make sense unless there's some kind of invisible supply line keeping everything alive.
And that supply line is exactly what we're going to unpack today.
Exactly. Because it turns out that supply line is a microscopic, solar-powered engine hidden deep inside the coral's own cells. And for this deep dive, we're looking at a groundbreaking 2026 paper from the journal Cell by Maruyama and colleagues, which finally explains how this works.
Yeah, and their mission and ours today is to really get into the exact molecular mechanisms of this, how corals and their relatives, the sea anemones, literally hacked their own cellular biology.
They turned themselves into part plant, part animal hybrids, right?
Precisely. We're going to explore this decades-old biological debate, the cutting-edge cell sorting they used to solve it, and the genetic conveyor belt that keeps the whole reef from collapsing.
So let's start with the basic setup, because the context here is just fascinating. You've got these corals and anemones, and they capture these microscopic algae from the water.
Right, dinoflagellate algae from the family Symbiodiniaceae.
Yeah, try saying that 3 times fast.
Oh.
But the animal host literally swallows this algae, right? Through a process called phagocytosis.
Exactly, it just engulfs it. And the algae gets pulled into these specialized host cells, which we call symbiocytes.
Okay, symbiocytes.
Right. And once inside, the algae is housed in this custom-built cellular compartment known as the symbiosome.
And this symbiosome, this little compartment, this has been the center of a massive scientific argument for, like, what, decades?
Oh, absolutely, decades. Biologists have been tearing their hair out trying to figure out what the symbiosome actually is. So there were 2 main camps.
Okay, what was the first camp?
One camp argued it was an arrested phagosome.
Arrested phagosome, meaning what exactly?
So think of a phagosome as just a temporary transport bubble. The host cell swallows the algae, wraps it in a membrane, but then the normal digestion process just magically pauses halfway through. It gets arrested.
Got it, like a holding cell. And the other camp?
The other camp argued that the symbiosome was a phagolysosome, and that is the formal term for a fully developed, highly acidic cellular stomach.
A cellular stomach,
Yeah.
Okay. But proving which camp was right, that sounds like it should be easy, but it was actually a technical nightmare, wasn't it?
Oh, an absolute nightmare. Because to study the symbiosome, you have to extract it perfectly intact.
Right, and I was thinking about this, like, imagine trying to pull a highly sensitive engine out of a car, but you can't bring any of the chassis, none of the wiring, no oil, just the pristine engine.
That is a perfect analogy. And doing that with stony corals is nearly impossible because they have these hard calcium carbonate skeletons. Extracting pure cells from rock is, well, it's a mess.
But what did Maruyama's team do instead?
They turned to a model organism. They used a specific sea anemone called Aiptasia.
Ah, okay. Because anemones have the same symbiotic relationship with this exact algae, but they're soft-bodied.
Right. So the team could take the adult anemone and completely dissociate it into this soup of single cells. But that creates a new problem, a massive sorting problem.
Because now you just have a smoothie of microscopic parts.
Exactly. You've got a flurry, you have intact host symbiocytes that still have algae inside, you have free-floating symbiosomes that popped out, and you have naked algae that lost their membranes completely.
So they needed a way to physically isolate just the pristine symbiosomes from that debris. And they use a centrifuge for this, right? Spinning it to separate things by weight and density.
They did, but just spinning it isn't enough. Pulling a layer out of a centrifuge doesn't guarantee you have the symbiosome. You might just have loose algae.
So how did they prove what they actually had?
This is where it gets brilliant. They designed a 3-color visual security system. First, they used a dye called calcine violet.
Calcine violet, what does that do?
It stains the host animal cytoplasm purple.
Yeah.
So any leftover animal cell gunk glows purple. Second, they used a custom antibody called RHBG.
Okay, RHBG.
Right. And this antibody was designed to lock onto a specific protein found only on the outer membrane of the symbiosome itself.
Got it. And the third color?
They just use the algae's own natural chlorophyll, which autofluoresces bright yellow under the right light.
Oh, that is so elegant.
Yeah.
So when they run this through a flow cytometer, a laser sorting machine, they can see exactly what they have.
Exactly. If you see purple, you know you've got host cell contamination. If you see just yellow, you're looking at naked algae.
But the jackpot, the perfect car engine, is a yellow glowing core tightly wrapped in that RHBG antibody with zero purple.
You nailed it. And by refining this, they hit a staggering 80% purity. 8 out of 10 of the algae they isolated were perfectly encased in symbiosome membranes, entirely stripped of the host cell.
80% purity. I mean, in cell biology, that is just a monumental achievement.
It really is. It was the bottleneck holding the whole field back.
Okay, so let's do a quick mini recap before we dive into what they found. So point 1, corals house their algae in these little organelles called symbiosomes.
All right.
Point 2, scientists were fiercely debating if this was just a safe transport bubble or an actual cellular stomach.
Exactly.
And point 3, using a soft-bodied anemone and this totally novel color-coded sorting technique, researchers finally extracted pure symbiosomes to study.
That's the foundation, yes.
So I have to ask you, the listener, right now, think about this. If you were the host cell, would you risk putting your precious solar-powered algae inside a highly acidic stomach? Because that sounds like a terrible idea.
It sounds suicidal for the symbiosis, honestly. But that paradox is what makes the proteome data so shocking.
Right, because once they had the pure symbiosomes, they cracked them open and mapped the proteome, every single functional protein making up that structure. So what was inside?
Well, first they look at the membrane, and it was heavily populated with membrane trafficking proteins called snares.
Snares, like a trap.
Kind of. In cell biology, snares act like mechanical grappling hooks. They stick out of a membrane, grab onto passing vesicles, and force them to dock and fuse.
Oh, wow. So the symbiosome isn't just an isolated bubble. It's actively reaching out and interacting with the rest of the cell.
Actively interacting. And what vesicles are these snares grabbing? They're grabbing lysosomes.
The cellular stomachs.
Yes. The proteome was overwhelmingly dominated by lysosomal machinery. The debate is over. The symbiosome is undeniably a phagolysosome.
So it is a fully functional stomach.
100%. They found massive amounts of this complex called V-ATPase.
V-ATPase. Okay, what is that?
It's this aggressive molecular pump. It burns energy to literally shove protons across the membrane and into the compartment, and that sheer volume of protons drops the internal pH down to around 4.
Wait, hold on. A pH of 4? That's roughly the acidity of tomato juice or even like acid rain.
It is extremely hostile.
If you plunge a single-celled algae into an environment that acidic, its internal chemistry should just start breaking down, right? Proteins denature at that pH.
They do. And the hostility doesn't even stop at the acidity. They also found heavy concentrations of structural proteins like LMP1B.
LMP1B, what's the function there?
Its primary job is to heavily coat the inside of a lysosome, basically armor plating it, to prevent the cell's own acid and enzymes from eating through the membrane and destroying the host from the inside out.
Okay, so the host is armor plating this compartment because it knows how dangerous it is inside.
Exactly. But the most definitive evidence was the cathepsins.
Cathepsins, those are the actual digestive enzymes, right? The molecular scissors.
You got it. The proteome showed super high levels of cathepsin B, D, Z, and L.
That's a lot of scissors.
And here is the crucial detail. The mass spectrometry didn't just find the genetic blueprints or the inactive, safe precursor forms of these enzymes. It found peptides belonging to the mature, fully activated forms of these cathepsins.
So safety caps were completely off.
Completely off. These enzymes were in there actively cleaving proteins.
And they visually confirmed this too, didn't they?
They did. They double-checked with a live cell dye called lysotracker green, which only lights up in highly acidic environments, and the symbiosome just lit up perfectly. They even caught images of smaller lysosomes actively fusing with the symbiosome, dumping in more acid and more enzymes.
Okay, I have to stop and push back on this, because the mechanics here seem fundamentally broken. Well, we have a highly acidic stomach, a pH of 4. It's pumped full of mature, active digestive enzymes. If the safety caps are off and the scissors are cutting, how on earth is the algae surviving? Why isn't it just dead and digested?
That right there is the paradox that makes this whole thing so remarkable. The algae isn't just passively sitting there taking the assay. It has this highly specialized defense mechanism.
Okay, what is it?
Earlier structural studies showed that these specific Symbiodiniaceae algae have multiple complex layers of membranes between their main cellular body and the host's acidic stomach wall.
Oh, like a shield.
Yes. And they generate these layers through a continuous shedding process called extisis.
Exitisis. Now, when I hear exitisis, I immediately think of like a snake shedding its skin, just slipping out of a dead, useless layer. But at a molecular level, shedding a membrane is a huge energy cost.
Massive.
Because membranes are made of super valuable lipids, carbohydrates, and proteins. Why throw that away?
Because that energy expenditure is the whole point. The algae are intentionally pushing out these rich lipid and carbohydrate membranes into the highly acidic space.
Wait, they're feeding the stomach.
Yes. The host's mature cathepsin enzymes are actively attacking and digesting those shed outer layers.
Oh, wow. So the host isn't actually trying to eat the algae itself. It's harvesting nutrients by digesting the protective walls the algae keeps throwing up.
Exactly. The algae is effectively paying rent with its own cellular membranes.
That is wild. It stays perfectly safe inside its innermost layers, just soaking up sunlight, making energy, while the host's stomach constantly digests the discarded trash layers.
It completely shifts our understanding of the relationship, doesn't it? The host gets a steady supply of energy-rich molecules right across the symbiosome membrane, and the algae avoids getting completely digested. It's this highly calculated, biologically managed negotiation.
Okay, so let's pause for another mini recap here, because this is dense.
Yeah, let's do it.
Point 1. The symbiosome is undeniably a lysosome, a cellular stomach. Point 2. It is loaded with Aiptasia acid pumps and active mature cathepsin digestive enzymes. It is a hostile environment.
Very hostile.
And point 3. The only reason the algae survives is by constantly throwing out these sacrificial membrane layers through ekitasis, which the host then digests for nutrients.
Spot on.
So listen, I want you to think about how this dynamic shifts our view of symbiosis. We always picture this peaceful Disney-style partnership, but this looks way more like a heavily armed standoff where both sides are just extracting exactly what they need.
Right, but a standoff only works if both sides have something valuable to trade. We see how the host extracts its value by digesting the shed membranes, but the host also has an obligation.
Right, the algae needs something in return.
Exactly. If that algae is going to run its internal solar factory and keep producing those costly membranes, it desperately needs raw materials. Specifically, it needs a massive, uninterrupted supply of inorganic carbon and sulfate to run photosynthesis.
And here is the fatal flaw for the algae. If it's trapped inside a sealed acidic stomach wrapped in multiple layers of host membrane, it is completely cut off from the surrounding ocean.
It's totally isolated.
It can't just absorb carbon from the seawater.
Yeah.
The host has to actively pump those raw materials into the acid bath, or the whole system just starves to death.
And this is where the proteomics data provided another huge breakthrough. When the team sifted through the thousands of proteins on that symbiosome membrane, they zeroed in on one very specific lysosomal transporter called SLC26A11.
Okay, SLC26A11. I look at this thing as the carbon conveyor belt.
That's a great way to picture it.
You have this massive physical barrier between the algae and the ocean, and SLC26A11 is this dedicated pipeline embedded right in the stomach wall, actively feeding the raw materials for photosynthesis straight into the algae's factory.
Exactly, it's the pipeline. But, you know, in biology, just finding the protein in a specific location doesn't actually prove what it's doing.
Right, you have to break the machine to see if it stops working.
Precisely. And the validation process they used for this was just incredibly rigorous. They did a two-stage genetic disruption.
Okay, what was stage one?
First, they went back to their model sea anemone, Aiptasia, and they utilized CRISPR knockdown technology.
CRISPR. So, without getting too deep into the weeds for everyone, CRISPR basically allows scientists to put in synthetic RNA molecules that intercept and destroy the messenger RNA for a specific gene, right? Before it can even be translated into a protein.
Exactly. It acts like an interceptor. So they used it to suppress the production of the SLC26A11 conveyor belt. And when they did this, the anemones showed a rapid and significant drop in their symbiont density.
Because the algae were effectively starving and dying off.
Right. The pipeline was shut off.
Okay, so that proves it in a lab model,
Yeah.
but the leap they took next is what makes this a landmark paper, in my opinion.
Oh.
They didn't stop at the anemone.
No, they didn't. They moved to an actual wild reef-building coral called Galaxia fascicularis.
And doing genetic editing on wild coral is just a logistical nightmare.
Oh, it requires an immense amount of precision and timing. Corals generally only spawn once or twice a year, releasing their eggs and sperm into the water column in this highly synchronized event.
Just a massive underwater blizzard.
Yeah. So the researchers had to wait for this exact spawning window. They collected the gametes, let them fertilize in the lab, and then they had a painfully short window of time to work with these single-cell zygotes before they started dividing.
You literally have hours to pull this off.
If that. So within that tiny window, they micro-injected those single-cell coral zygotes with CRISPR-Cas9 machinery.
Wow.
And they programmed the CRISPR complex to specifically hunt down that SLC26A11 gene and permanently mutate it, physically breaking the DNA sequence so the coral could never, ever produce that carbon conveyor belt.
I mean, doing CRISPR on a single cell of a wild organism that only spawns once a year, the pressure there is just wild.
The stakes were so high, but the physiological results were incredibly stark.
What happened?
When those CRISPR-mutated corals started developing, their algal density just plummeted. Their entire physiology was severely impaired. By snapping that carbon conveyor belt, they completely shattered the symbiosis.
Okay, let's do our third recap. So point 1, SLC26A11 is a crucial transporter, the conveyor belt providing carbon and sulfate to the algae.
Right.
Point 2, disabling this transporter completely ruins the symbiosis.
It breaks the whole system.
And point 3, they definitively proved this using CRISPR gene editing in both a model anemone and a wild reef-building coral.
Yes. And proving this mechanism in both a model and a wild stony coral shows this isn't just some weird anomaly. The SLC26A11 transporter is a deeply conserved foundational mechanism that underpins the entire reef ecosystem.
Let's reflect on this pipeline for a second, because the elegance of it is striking. We know that if you genetically disable this conveyor belt, the algae starves, everything collapses. But this raises a huge evolutionary question for me. Well, we know that this kind of intracellular photosymbiosis, an animal housing an algae inside its cells, has evolved independently multiple times across completely different branches of the tree of life, right?
Oh, yeah, many times.
So how did nature pull off this incredibly complex, highly specific engineering trick so many times? It seems almost impossible that so many different animals would just randomly invent a customized, solar-powered organelle from scratch.
Right, because building a brand new organelle from scratch would be an evolutionary nightmare. It takes way too long. But the beauty of this paper is that it provides a very clear, logical model for how this evolved, and it relies entirely on the sequence of events.
Okay, walk me through the sequence.
Step 1. An early organism encounters an algae and eats it, just through normal phagocytosis.
Just looking for a snack.
Exactly. The algae is sent to the cellular stomach to be digested. But step 2, because the algae has that ecdysis trick, because it can shed its outer layers, it actually survives that initial acid bath.
Right, it pays rent and stays alive.
And this is where the evolutionary shortcut happens. The cellular stomach isn't just a dead end. In any normal cell, the lysosome is already the primary nutrient distribution center.
Oh, because it's breaking things down and sending the nutrients out.
Exactly. All the cellular highways, the recycling machinery, the transport proteins, they are already routed directly to the stomach.
So the plumbing was already fully installed.
Yes. The host organism didn't need to invent a brand new compartment. It didn't need to build new highways to move carbon in or sugars out. All it had to do was co-opt its existing lysosomal machinery.
It just repurposed what was there.
Right. It took proteins that were already sitting on the stomach wall, like our carbon conveyor belt SLC26A11, and just slightly tweaked their regulation. So instead of just contributing to digestion, they started actively trading nutrients with the algae.
That is the ultimate biological hack. It provides this really simple evolutionary path for an incredibly complex relationship. The animal kingdom didn't reinvent the wheel, it just repurposed its digestive tract.
Co-opting existing biology is a huge theme in evolution, but we very rarely get to see the exact molecular gears laid out this clearly.
No, we don't.
Seeing the V80 passes, the cathepsins, and the SLC26A11 transporters all working in harmony to maintain this really delicate balance between digestion and starvation. It's a massive leap forward in our understanding of cellular biology.
So why does all this microscopic plumbing matter to you, the listener? I mean, why should we care about the exact proteome of a coral cellular stomach?
That's a fair question.
But the answer is that this level of detail is the absolute foundation for understanding and potentially saving coral reefs. Because we are in an era of unprecedented environmental stress, right? We're facing ocean warming, acidification, and that leads to these mass bleaching events.
Right, where the corals basically panic, expel their algae, and starve to death.
Exactly. And for a long time, we've only been able to observe bleaching at a macro level. We just see the whole reef turn white. But if we don't know exactly how the underlying engine works, We have no idea how to fix it when it breaks.
Exactly. But now, because of Maruyama's research, we have the schematic. We know the exact parts.
We know that if thermal stress disrupts that VATPase acid pump, the whole negotiation fails.
Right, and if ocean acidification interferes with the SLC26A11 carbon conveyor belt, the algae starves and the entire metropolis collapses.
So by monitoring these really specific molecular pathways, scientists can pinpoint the exact biochemical failure points of bleaching.
They can better predict which coral species possess the robust genetics needed to survive a changing climate, and maybe, just maybe, find ways to help them adapt. It transitions our whole approach from just observational ecology to targeted molecular intervention.
That's incredible. So before we wrap up, I want to leave you with a provocative thought to mull over on your own. We just walked through how nature repeatedly created these intricate solar-powered partnerships simply by tweaking a cellular stomach, right?
Right.
It took an existing lysosome, added a few nutrient conveyors, and built a part plant, part animal hybrid. So my question is, if evolution can hack a stomach to create a solar-powered animal, could we?
Oh, that's an interesting thought.
I mean, as our mastery of CRISPR and cellular engineering grows, could we one day use this exact blueprint of co-opted lysosomes to engineer entirely new artificial symbiotic relationships?
Synthetic biology.
Exactly.
Yeah.
Could we design synthetic biological machines or even, new types of agricultural crops that feed themselves by housing customized solar-powered algae right in their own modified stomachs?
When you look at the mechanics laid out in this paper, engineering artificial endosymbiosis starts to look a lot less like science fiction and more like just a complex engineering challenge waiting to be solved.
It really does. So next time you see a picture of a bustling neon coral reef thriving in a complete ocean desert, remember the microscopic stomachs working overtime to make it all possible. Keep questioning the invisible mechanics of the world around you.